When you look at the Pfizer news this week, are we putting too much hope on this? What are the main concerns that you have? Is it how you distribute it, or actually just that the trial will mean a vaccine in the next 2 to 3 months?
Well, first, it's good to be optimistic, and certainly the release, the press release, gave us very good numbers for the initial vaccine efficacy studies with that vaccine. I think Pfizer is the first of several of the same kind of vaccines, so that also bodes well for some of the other clinical trials that are going on. I think it's always the granularity of the data. We have to realize that it's very different to vaccinate children versus adults versus the elderly. So we don't know any of that information in terms of how well different groups were responding and being protected. And I think the issue with Pfizer that really comes through is in terms of distribution, becomes the cold chain that's needed. That vaccine needs to be stored at very cold temperatures to ensure long-term stability, and that infrastructure may be a challenge in terms of distributing the vaccine eventually.
What do the mink farms in Denmark actually tell us about how this virus is changing?
Yeah, so it's another important question. We haven't seen the firm scientific data yet, but it seems like the virus has made a jump into the mink population from humans. That's actually occurred in multiple countries around the world. But the unique thing in Denmark is that they've had some transmission in the mink population, and the virus has then come out of minks and back into humans. So that's what we call a reverse zoonosis, an animal virus, a human virus going into animals and then coming back into humans. The important thing, though, is that virus has a couple of changes in its surface protein that appear to affect the ability of antibodies to bind to that. So it'll be very important for us to really keep monitoring that outbreak in Denmark to see if this virus really is something that's a little bit different. The analogy I use is influenza, where we have seasonal changes in the virus that force us to update the vaccine on an annual basis. So this may be an example of the virus changing in that way. Denmark is using culling of the animals to really control the outbreak, and we'll have to keep monitoring the virus to see how fit that virus is to spread in the human population and how much it really has affected its overall, what we call antigenic structure.
Andrew, can you give me a sense of, once we have the formula, the correct formula, and the vaccine starts being distributed, there will be a list of priorities, right? If you're of, at risk or a certain age group, when can it be widely available for everyone?
That's going to take some time. There are two things that limit that widespread distribution: the availability of vaccine. The vaccine manufacturers have gotten a head start in many cases in terms of generating their vaccine. But this vaccine requires 2 doses, and that, of course, makes a very big hit in terms of how many people can be effectively immunized. You'll see a scaling up of populations. We'll probably be focusing on frontline responders, healthcare workers, as well as some of the parts of the population that are very susceptible to the severe disease as the first group of individuals that are getting vaccinated. But you won't see widespread distribution of the vaccine to the general public until, I'm guessing, sometime in the middle of next year. Simply because right now we only have one vaccine that looks like it's going to get some approval soon, and it's going to be very limited in terms of the number of people that can be immunized for the first few months.
There are a number of questions, Andrew Pekosz, about testing and whether these are reliable. Positive tests, false positives, false negatives. Elon Musk tweeting about it a couple of hours ago. Does that, do these kind of debates undermine the credibility of what we know works?
Well, we know that a lot of tests have been rolled out under the emergency use authorization. And what that means is that there's signals that the test works well, but it certainly requires more testing to make sure that we have a broader sense of how well it works under very unique situations. So it's important to understand, number one, what test you're getting. Is it an antigen test? Is it a PCR test? And then it's also important to put that into context in that a test itself should never be relied on 100% in terms of providing you some protection. You've got to take the test in context. An antigen test may be borderline positive early in the infection, but by the time you start showing symptoms, it may be a really strong test at that point in time. So we have to understand that testing is not the be-all, end-all solution here. It's one part of our approach to this outbreak that can limit the spread of the virus, which, as everybody knows, particularly in the U.S. and in parts of Europe, is just incredibly widespread today.